Assalamu alaikum wa rahmatullah. Greetings. This is Dr. Bilal Wajid, and today I am teaching this course along with Dr. Faria Anwar. I am an electrical engineer from Texas A&M, and she's a medical doctor from the Shifa College of Medicine. This particular course, titled Biomedical Signal and Image Processing, will hopefully, using a medical doctor and engineer, should be able to give a holistic picture of the course. And that's the idea of having two faculty members from two different domains, because this is an interdisciplinary course. Apart from these two individuals, we have two teaching assistants, Tajdar Alam and Hafsa Rafiq, both of whom are graduate students at Government College University, who shall be helpful in developing worksheets, quizzes, assignments, and exams for this particular course. Now, this course is divided into three components. The first component is where we introduce signals and images. The second is discussing several biomedical signals, and the last component talks about biomedical images. In the first part of the course, we are talking about signals in general, one-dimensional signals, two-dimensional signals. For instance, an image is a two-dimensional signal, because it has both x and y coordinates. And whether they are defined in gray level or they are color images. And if you can see, the whole component is basically talks about image filtering. So if we're going to discuss low pass, band pass, high pass filters, Butterworth filters, and how those apply to images. Now, within image processing, there are several different transformations which are used to highlight certain components of the images and to fade additional or unrequired pieces of information. So the same goes for signals. In terms of signals itself, one of the first things we would discuss is Fourier transform, because it is one of the most used transformations that help convert a time domain signal into a frequency domain signal. And obviously with Fourier transform comes discrete Fourier transform, and we would be studying all of that. So the first component of the course will have, you could say, 6 chapters. Chapter 1 discusses signals and images, really the fundamentals of that. Then Fourier transform, image enhancement, restoration, filtering, chapter number 4, 5, and 6. Part 2 of the course talks about biomedical signals. Here is where we're going to talk about electrical activities of the cell, how the cell interacts with other cells, how the neurons work, for instance. How does the electrocardiogram, which is one of the principal diagnostic tool for cardiovascular diseases, how does that operate? What defines, and within ECG, obviously, you will be looking at several various types of waves and how you can differentiate one and another, and what does it mean if somebody has a high peak. Same for EEG and electromyograms. Now, part of this course, this component, which is part 2 and part 3. So in part 3, you can see you have the X-ray. Now we're talking about medical imaging and magnetic resonance imaging. I guess everybody knows about the X-ray, because I'm sure every family member has one man or one lady who has a broken bone in their body, or have broken a bone once in their life. So everyone is aware of the X-ray. But apart from the X-ray, we're going to talk about the magnetic resonance imaging and how does it help to look into diseases, neurological diseases. How can you look at the brain? How can you use ultrasound and PET scan? Now, part 2 and part 3 of the course is where both Dr. Faria Anwar, who's a medical doctor, and me, who's an electrical engineering PhD, both of us will come together and discuss these topics so that you have, not only are you looking at how you're going to look at an X-ray, or I'll give you the example of MRI. So Dr. Faria Anwar will tell how an MRI is looked, how is it useful, how does it relate to different diseases, why is it such an important tool. Whereas I would be talking about how you can develop software tools for MRI. For instance, there's one particular tool that we've developed which is called Exonica, and it basically is working on a Ubuntu-based operating system where you can basically help to develop software workstations. If you want to have a laboratory for an MRI and you want to teach bioinformatics students or biomedical students or students from computational biology how to use MRI software, so Exonica is a very good tool for that particular purpose. So this course is good, in fact, as an upper undergraduate level course, a 400 level course you could say, or it works well as a graduate level 500 or 600 level course in biomedical and image processing. The course can be intended for electrical engineering students or biomedical engineering students, but at the same time it can be a very good course for students whose majors are bioinformatics, whether that's taught in computer science or it's taught in life sciences, or even for students whose major is computational biology. So this course can be used by multiple programs for their teaching needs. The textbook that I would be following for this entire course is by Robert Splinter and Najarian, which is Biomedical Signal and Image Processing. It is a good book, and I've come to like it. The explanations are well, and I would recommend this book for this course. Now, a typical biomedical system contains the following 5 components. And this course particularly starts here. This is the point. In other words, we would assume that the sensor has provided us the image and we are going beyond this point. However, to be honest, we are talking about electrical activities of the cell. We are talking about different imaging techniques, different biomedical processes. So we will, in that process, discuss sensors as well. But part 1 of the course, which talks about signals and images and how you filter them and how do you extract important information, this is pretty much part 1 of the course. Now, one thing which comes as a cautionary note is, in my experience, a lot of people who are working in health informatics or biomedical signal processing, a lot of those individuals are not collaborating with medical doctors. They are not collaborating with people from veterinary sciences. And this, in fact, limits their potential into using their current knowledge and applying in these domains. I'll give you a very small example. For instance, we have a paper where we developed a diagnostic test for basically a disease of the bowel, and it's called inflammatory bowel disease, or IBD for short. Now, the reason, and that diagnostic test has been published, that diagnostic test has been licensed by a diagnostic company, IDEXX Laboratories. It's one of the biggest laboratories in the US. But the reason we were, you could say, successful in this process is because that problem was informed to us by the people in veterinary sciences, by the veterinary school of Texas A&M. And they have a gastrointestinal laboratory over there headed by Dr. Jan Szydolski. So Dr. Jan Szydolski, who is a veterinary scientist in one of the Texas's biggest, one of the biggest veterinary hospitals, now he was the one who informed us that here we need a diagnostic test and we can work on IBD because we are working in the gastrointestinal laboratory, and let's just work on inflammatory bowel disease. Now, because he was very clear on what the problem was, and he was very clear on the subjects, because again it's a veterinary hospital, so dogs and cats are coming in regularly. We as engineers were able to assist his mindset where he wanted to go with the knowledge that we had, and collectively it came out to be a pretty successful, I would say, endeavor. And within this course, we will be having a separate lecture on that. But from that particular example, from that particular exercise, we ourselves realized that a lot of people in this domain are doing research which they claim to be of biological relevance. What I say is, well, if you have 100 things which are biologically relevant, and if you can isolate them and identify them and determine them, only 1 or 2 or 3 of them are of clinical relevance. In fact, most of the things which are biologically relevant may not be clinically relevant. Therefore, your research work is extremely useful if you orient yourself to things which are of clinical relevance rather than biological importance. And for that to successfully occur, it is best that the problem and guidance be derived from people working in medical sciences. So I'm talking about medical doctors. If it's humans, if it's animals you're interested in, then it's veterinary sciences. So that was the introduction of the course. I hope we meet again. This is, you could say, part 0. Next lecture we're going to talk about signals, and thank you very much. Have a good day.